So here's the chain of events we want to look at, right? We have this single motor neuron coming down, branches off, gets to this motor end plate right here, right? There's going to be an action potential that comes down the length of the axon, ending up at the axon terminal. That is going to lead to this electrical signal being changed into a chemical signal at the neuromuscular junction. And that chemical signal is going to change back into an electric signal at the motor end plate there. We're going to look at how exactly, this is more of the summary about what happens. We'll look at each step here. So at this yellow region over here is the motor end plate, right? That's a region on the muscle fiber. This is here they're calling it the synaptic end bulb, right? Or terminal bouton, whatever. This is the junction or the synapse. And so in between the terminal bouton, the synaptic end bulb, and the motor end plate is the synaptic cleft, right? A little space, right? Neurons could communicate with other neurons very rarely through gap junctions, that is direct connection, and cardiac muscle does that too. But in this case, in most cases in the nervous system, and always at the neuromuscular junction, they don't quite touch, right? There's a little bit of a space in between your axon terminal and the muscle fiber. And that little space is your synaptic cleft. Let's look a little closer here at the impulse, right? There's going to be an action potential going down here, a one-way signal coming down the axon. The same way, you'll have this series of voltage flips going down, a little different because it'll be myelinated. Don't worry about it. But there's this electrical signal, action potential coming down the nerve fiber, right? As it reaches the end here, the terminal bouton here, the synaptic end bulb, is packed with all these voltage-gated calcium channels, just like we saw in the T-tubules, okay? So these voltage-gated calcium channels respond to this action potential, this change, this depolarization event, by opening up, and calcium is high on the outside, low on the inside. So calcium rushes into the synaptic end bulb over here, right? So now you got this inside of the terminal bouton flooded with calcium, right? Due to this electric signal. All right, so calcium is flooded in, and we don't get into the details of how it does that, right? All we say is that the entering calcium stimulates these vesicles in here, right? Remember we talked about vesicles and exocytosis, right? These little packets of, in this case, neurotransmitters, right? So these are filled with the neurotransmitter acetylcholine over here, acetylcholine, right, in red. So you have all these synaptic vesicles loaded up in here. They're just waiting to go. And calcium is that signal for it to fuse to the synaptic end bulb membrane over here, as exocytosis does, right? Fuse with it and release its contents, the acetylcholine, into the synaptic cleft, right? So voltage signal, calcium influx, some kind of magic with the interaction with the vesicles leads to a fusion of those vesicles, just like any exocytosis interaction, leading to the release of acetylcholine. Now we'll see acetylcholine is going to diffuse across the synaptic cleft. So here we go. Here's that exocytosis event over here. These little vesicles packed with acetylcholine, the neurotransmitter, the signal. You're going to fuse with the membrane here, release the contents into the synaptic cleft. The neurotransmitter diffuses across that very short distance here, right? It fills it up with here. And then you have the motor end plate that has these junctional folds over here that are packed all around with your acetylcholine receptors, right? So, you know, the release of neural signal is very, confined to this area, unlike your hormonal signals. And then the place that it releases is packed with receptors for this neurotransmitter. What happens at this point? The neurotransmitters, acetylcholine, binds to the acetylcholine receptor. Right? This is a ligand-gated ion channel, as we talked about. Upon the binding of acetylcholine, the receptor opens up, right? Because it's a receptor and an ion channel, ligand-gated ion channel, and in flows all the ions according to their concentration gates. We're just going to talk about sodium for the muscle, right? In reality, there's also potassium going through, but we'll talk about that when we get to the neurons, right? Right now, we'll just focus on sodium, which is high on the outside out here, and is going to rush into the muscle fiber. What is that going to do? It's a big bunch of positive charges over here, which is going to reach the threshold for all these adjacent voltage-gated sodium channels. Right? So you created this local change from negative to positive, which opens up these voltage-gated sodium channels. And that's going to create that action potential. So let's look what happens. This is actually a very short action. As it's released into there, it binds to the receptors, right? Opens up the sodium channels and causes this membrane potential. But again, the nervous system wants to be very controlled over the effect on its effector, right? It wants to have a very specific, short-lived response. So as it binds it, right, there's another thing going on, right? Acetylcholine is starting to get broken down immediately, right? And so it's broken down by this acetylcholinesterase, right? Acetylcholine esterase. That's an enzyme. What that does is break down the acetylcholine into the acetyl and choline groups, right? And that's not going to activate the receptors. Okay. So that means the response to this acetylcholine binding is pretty short, right? It opens up, and as soon as it releases it, this is the way these ligand channels, they often like just bind it, open up and release it. And then the acetylcholine is breaking down, right? Because you got this enzyme activity breaking down the acetylcholine. So it's a very quick, specific, short-term response. And if the neuron wants this muscle fiber to contract again, it's got to send another signal, unless something happened, right? So when we're talking about these synapses over here, this is often the site of like poisons and drugs and stuff like that, right? You want to have some kind of effect on the receptor ligand interaction, right? The neurotransmitter and the receptor interaction. So anybody here heard of Botox? Of course you have, right? Right. What does that do? It paralyzes the muscle. What Botox does, the botulism toxin, what it does is block the release, right? It blocks this exocytosis event, and it prevents this. So, and that way the muscle is paralyzed, right? It doesn't get any input from the motor neuron. So that's one way this drug works. It blocks the release of neurotransmitter. Other drugs might block this receptor so that it blocks it, but it doesn't open it. And then acetylcholine can never bind to the receptor, right? That would act as an antagonist to acetylcholine, and that would also paralyze you. Other poisons or drugs are going to inhibit the effect of this acetylcholinesterase activity. And in that case, the opposite effect happens. You have too much acetylcholine in this region right here, and the muscle keeps on contracting when the nervous system doesn't want it to. And so all these kind of things can mess with this interaction in one way or another. There's other stuff, right? Once there's other mechanisms to get rid of this neurotransmitter in the nervous system, is to take it back into the cell up here, right? Reuptake it. Like, so if this was serotonin, we have serotonin reuptake inhibitors, right? Those are things like Prozac and stuff like that, so that serotonin remains in your synaptic cleft longer. It makes you feel happy somehow, right? Those are like antidepressants and stuff. So we'll get to this when we get to, neurotransmitters and stuff, but just while we're here, this is many most of the like drugs and stuff we talked to, these neuroactive drugs, a lot of poisons that deal with the muscles, there's some kind of interaction somewhere along this line right here. All right. So acetylcholine is broken down by acetylcholinesterase. And so you don't get a signal. And further down the line, right, when you look down at the release of calcium, calcium is pumped back into the sarcoplasmic reticulum. And then there's no longer calcium, and that interaction between myosin and actin ends, right? And then that's when the muscle relaxes. So that's the end of it right there. One more thing. A skeletal muscle fiber has this single muscle cell, muscle fiber has one single neuromuscular junction, right? Usually along the midpoint of it. Okay. If you think of your longest muscle of your body, right, which is your sartorius, usually that muscle that goes over the front of your leg, like a strap-like one, goes from your lateral to your medial side on the front of your thigh, right? That could be over 2 feet in some people. It has a single neuromuscular junction somewhere in the middle of it, right? And so when that neuromuscular junction is depolarized, that signal is going to spread in both directions, right, throughout the entire thing, so that the muscle fiber contracts simultaneously. Okay, so there's only one neuromuscular junction per fiber, right? We'll get back to this concept when we talk about motor units, right? But for now, right, when you're looking at a chain of events, you have the nervous cell right here, the motor neuron, sending a signal down the axons. They're going to release. Here's the acetylcholine, right? And then there's that signal going throughout the length, right? And then electrical signal will release cause a calcium release, so on and so on. Right? So again, it's going to spread from a central point, whatever the neural middle of the muscle cell, and spread throughout the length of that. Right? In neurons, we'll see the ion channels are arranged in such a way that the signal is one way. It only goes in one direction, right? Toward the axon terminal. All right, but in muscle cells, it's going to go down the entire length. All right. So let's look at the whole sequence of events, right? I kind of told it all out of order. And you want to think of this. This is the advantage of this unit, right? You could think of things in a sequence of events rather than, on the other 2 units, we had things like all over the place, right? And you had to kind of categorize things in your mind. Here, we want to look at a sequence of events that happens, right? The action potential in the muscle fiber, and then in the neuron, sorry. And then you just want to remember the names of everything. Vesicle release, acetylcholine, ion channels open, action potential, down the T tubule, right? We're going to release calcium over the sarcomeres over here, right? Calcium is going to be released over it. Calcium is going to be the signal for your interaction between your myosin and actin filament to contract. Right, so here's the summary picture. I don't think I put this on here, but it's a figure from your book, right? Just be able to kind of follow this event. This is the easiest way to think about it, a chain of events. Right? Just follow this nerve action potential, acetylcholine, right? Action potential, down the T tubule. What's happening here? Voltage gated calcium channels, calcium release channel, calcium flooding over the sarcomere, right? Calcium exposing the troponin on the thin filament. Exposing the myosin binding actin sites, elevated calcium released to contraction. Right? Once that contraction is done, calcium ATP pumps, put all that calcium back inside your sarcoplasmic reticulum, so that interaction ends and the muscle relaxes, right? So go over this verbally in yourself. Remember about learning and about trying to remember stuff. If you say it verbally, that's building neural circuits in your head. If you draw it out like this by yourself, as much as you can by memory, that's building neural circuitry in your brain to help you remember this. Okay. And just remember all the terminology that goes along with it. As you find yourself, because you look at this picture and it looks fairly easy, actually, right? Okay, I get that, right? But say it and see what gaps you're missing. Especially if you have any kind of study group, or if you know anybody who knows anything about it, or just try to explain it to any very patient loved one. And if you can explain it to your 5-year-old, you're doing really good. So that's the whole sequence of events. And then the one thing we're missing here talking about is that whole role of ATP in the myosin-actin interaction, right? Remember, starting from the point of after the cross bridge formation and power stroke, the ADP and phosphate group was released, right? And ATP, a fresh ATP molecule, was bound to the ATP binding site on the myosin head. What that did was allow the myosin head to be released from the actin filament, right? So now it was released, right? And then it had enzymatic activity, ATP ACE activity on the myosin head and the ATP site, which hydrolyzed, right? Took that energy that was stored in that triphosphate bond, made it into an adenine diphosphate. Then it used that energy, stored that energy as potential energy in the cocked sort of orientation of the myosin head. Then when calcium comes, the myosin head could bind to the actin. When it's in that position, as soon as it binds to the actin, it's going to cause that power stroke, right? Moving it over there. As it does so, it's releasing the phosphate and ADP, leaving that open for the next step, right? And so this is going to happen over and over again until this little myosin head right here goes down the entire length, as long as there's calcium available for the exposure of these sites, and as long as there's ATP to release it from that site right there. All right, does everybody kind of get how you're going to go through the signal, right? This is the kind of one part of it that's slightly separate that you have to kind of, okay, here's a little side note on what's happening at this area right here, right? All the rest of the stuff you want to follow in a sequence of events from, the excitation, contraction, coupling. This is the whole role of ATP in this interaction. But that's it. That is how excitation at the neuromuscular junction leads to contraction of the sarcomere, leading to creating tension of the entire muscle. So next time we're going to start to look at how tension is created and the factors surrounding that. All right, see you next time.